I'm assuming loose familiarity with functional MRI data and what it looks like and basic statistical intuitions, but hopefully nothing more than that. Okay. So I'm going to talk about why would you use functionally defined regions of interest in functional MRI data analysis? That's the question. So first, let's start with standard group analyses. Okay, so the goal of a standard group analysis is to be able to say something that's generally consistently true in most people, right? So the goal is to do a statistical test on your brain-based data to say what is generally true across this batch of subjects, rather than just found in one or two subjects. Okay, what is the general case? Okay, the problem with this method is that different brains have different shapes. Okay, so we have a real problem in saying what counts as the same place in different brains. Okay, so different brains are shaped as differently as different faces are. And if you did your best to align two different faces, you still wouldn't do very well. And if you think about it, if somebody asked, is the freckle on my nose in the same place as the freckle on your nose? It's actually not a very well-defined question because our noses are slightly different shapes. So what exactly would that mean? Okay, so you could make stuff up. You could define coordinate systems for the nose. You could probably do better than nothing, but there's a problem in there. There's a vagueness at root when specifying the location of two different things that have different shapes. Okay? All right. So the standard solution to this problem is to just forge ahead undaunted, take a bunch of different brains, align them as best you can with a whole set of complicated algorithms that I will just skip over. You can stretch them and twist them on a number of axes and try to get them aligned as best you can. Okay? And then to set up a coordinate system in that space. Sometimes these are called Talairach coordinates or M&I coordinates. There's a bunch of different systems. They all share the property that you take this common space that you align all brains to, and you set up X, Y, and Z in that space so that you can specify the location in any brain in this coordinate system. Okay? All right. So now, in this coordinate system, we can define what counts as the same place. We define a voxel, a little three-dimensional cube in that brain here. This is shown abnormally large, so you can see it. It would actually be very small. And that voxel has an address of coordinates X, Y, and Z in the brain, and you could find it in any brain that was aligned to that common space. Okay? All right. Now we can run statistics across subjects. Skipping all the details, just statistics that look at that voxel in each of these brains and ask, what is consistent across subjects in the response of that voxel? Okay, everybody got the idea? Okay, now this is not a dumb idea at all. It's a perfectly reasonable thing to do, and sometimes it works. And when it works, hooray. Okay? Okay, but the problem is it can go awry in a bunch of different ways. And as I just mentioned, the central problem is you can't perfectly align two things that have different shapes. It just can't be done. Okay? Okay, so I said it's kind of like trying to align different faces. You do your best, you still get somebody's mouth on top of somebody else's chin. Okay? Or another analogy that Rebecca and I used in a paper quite a while back is the Gulf Stream. Okay, so mostly I just love this picture. It's nice and colorful, but we'll use it as an analogy. Here's the Gulf Stream as seen from space. The color is coding temperature. The Gulf Stream is this kind of river of warm water that comes up from Florida and comes up along the eastern seaboard, heads out into the Atlantic, and then goes across the Atlantic to Europe. Okay? And there are all kinds of interesting phenomena that oceanographers like to study about the Gulf Stream, the organisms that live in it, the chemistry of it, and so forth. The problem with the Gulf Stream is its exact path varies from year to year, in fact, from day to day. And that means that if you want to study the organisms that live in the Gulf Stream or the chemistry of the Gulf Stream, it doesn't work to just go to a certain latitude and longitude. Because sometimes that latitude and longitude will have Gulf Stream flowing through it, and sometimes it won't. Okay? And so what oceanographers do about this is go to that latitude and longitude and then measure the water temperature to find out if the Gulf Stream is flowing through there or not. And if it is, you can take a sample and answer a question about the Gulf Stream. And if it isn't, you better get back on your ship and move over and go find it first. Okay? Everybody get the analogy here? Coordinate systems don't capture things that don't map perfectly and consistently onto that coordinate system. Okay. All right, so is this merely a hypothetical problem? No, it's not. It's a real problem. So here is a kind of striking example. This is a slice through the brain like this. This is the back of the head, occipital lobe. This color code shows the percent of subjects that overlap in V1, in primary visual cortex, in a common Talairach coordinate system. Okay, and the color code shows that this is 10 subjects. This is based on post-mortem analysis of the actual structure of the brains. Doesn't matter. The point is that in this common space, you'll get only a tiny bit of brain in which all 10 subjects have V1 or primary visual cortex in that location. So if you wanted to study primary visual cortex, this would be a lousy way to do it, right? It just wouldn't work very well. Okay, now there are other ways to align it where if you actually take the folds and wrinkles into account, you can do actually extremely well, but this standard method of just aligning whole brains doesn't work well for many regions, and here's an extreme case. Okay, so why does that matter? The first problem is you might do a group analysis and find nothing at all in your group analysis, even if that thing you were looking for was present in every single subject, nearby, but just not perfectly overlapping. Okay? So here's an example from a paper that we're writing right now in my lab. This is the data from Rosa Leifer-Souza, and this is the bottom of the right temporal lobe, oriented. How is this oriented? Okay, this is the back of the head, front of the head, so it's oriented like that, looking at the brain this way. Everybody got this picture? The purple bits are the face-selective bits in these, I think, 13 subjects. And you can see every subject has like nice face-selective bits in roughly the same place on the lateral side of the fusiform gyrus. But when every single subject has that. But when Rosa puts these data into a standard group analysis, she finds no face-selective activation at all. Boom, nothing. That's a grotesque problem, right? You really miss things when you do that, okay? We've also tried this using other fancier kinds of registration methods that look at the whole cortical surface, and they didn't work much better than that, okay? All right. Now, it doesn't always fail. In these same data, the other colors are regions that show selective responses to color and to places, and both of those come through with the group analysis. So sometimes a group analysis works if the stuff happens to align right in that common space, and sometimes it doesn't. If it does, great. If it doesn't, you don't know what it means, okay? Okay. And in fact, because of this problem, I never would have found the fusiform face area in the first place had I had anybody to show me what I was doing. When I first started, I had no one to help, and so I invented all my own analyses, and I had no idea that group analyses even existed. So instead, I did a whole bunch of individual analyses like this and stared at them for a long time and said, everybody has something right there. So the human eyeballs work better than the standard statistical methods. Okay, but here's the second problem. You can find what looks like overlap of two different activation profiles in a group analysis that isn't present in any single subject that went into that analysis, okay? So here's a kind of diagram of how this can happen. We can take some set of mental functions and look at them in a group analysis, and you might get something kind of like this. Very overlapping activation patterns. Obviously, this is schematic. But you can get those kinds of results from a set of subjects, none of whom shows any overlap at all. And the way that can happen is each of these regions is in a slightly different location across subjects. There's anatomical variability that smushes and blurs out the activation. It makes these things look like they overlap when in fact they never overlap, okay? All right, so that's the second problem. And that happens too. This kind of mistake of claiming that things overlap when they don't is rare in people who study visual cortex, basically the whole back third of the brain, where there's a much more, I would say, serious approach to data analysis based, I think, in part on the fact that from the first days of functional MRI studies of visual cortex, people had lots of information to work on from prior neurophysiology work in monkey brains. And so they actually knew about distinct areas of visual cortex. And so they realized they had to find them first before characterizing them, not just smush them all on top of each other and hope for the best, right? When you go anterior of here, there's more of a problem, and people sometimes make this mistake of claiming that there's overlap when there's not. Okay, and you see that a lot in work on language and music, and a whole set of papers on the visual word form area by Kathy Price and her colleagues, all of which argue that visual word processing there's no selective region for visual word processing, all based on group analyses. Okay. Related to this, sort of corollary or flip side of the same problem, you underestimate the selectivity of a region in a group analysis, because you're averaging in from some subjects you're right on top of that region, and other subjects you're not. And if you average that stuff together, it will look less selective. Okay, so that's all that says. Okay, so these are the standard problems with group analyses. Again, I'm not saying don't do them. I'm saying they have a use. If you find something, great, but know what their strengths and weaknesses are. First problem is that you can miss things in a group analysis that are present in each subject. Second one is that you can see overlap in a group analysis that's not present in any individual subject. A third problem is more abstract, and that is this fundamental problem that if you read one study that publishes a result, okay, this is Joe Schmo's study published in one journal, and they find some activation right there, and they give you its Talarat coordinates. And another study publishes a result that's very nearby in Talarat coordinates, and they find something else. What do we do with this? How do we relate these two to each other? In science, we don't want each paper to be an isolated universe. We want to relate them to each other and build a consensus on what the actual correct story is, right? And we don't know how to relate these unless we believe that Talarat coordinates are, deep realities that we want to relate to, but we already know that they're not going to be. And so we have this really dysfunctional situation in a lot of the literature where we can't relate these different findings to each other. We do not have a cumulative research enterprise. Okay? So that's, I think, actually maybe the biggest problem with over-reliance on coordinates in group analyses. Okay, so enough of the problems. What do we do about it? There are lots of things that can be done about it, but the thing I want to talk about today is the solution that uses functionally defined regions of interest. Okay? So the idea here is, as I said before, if you want to study the Gulf Stream, you don't go to a particular latitude and longitude and measure the water there and just be confident. You go to that latitude and longitude and you measure the water temperature. Okay, we want to do the same thing in the brain. You want to study a face region, you don't go to a particular set of coordinates defined in Talarat or M&I numbers. You look in that vicinity and you see if that region is face selective. Okay? So the way you do that is you do something called running a functional localizer scan. You just take 5-10 minutes, whatever it is, scan a bunch of subjects on faces and objects, find the bit that's face selective in that subject. Okay? Write down those coordinates. Now you know where it is. Now you can ask it more interesting questions. Okay? All right. So, for example, here's some ancient data from my first paper on the fusiform phase area. We compared faces to objects. We get this nice little activation in a horizontal slice of the brain right there. We check by looking at the actual magnitude of response, and you see high responses when the person was looking at faces, much lower when they were looking at objects. That's our localizer. We found that region. Now we want to ask other questions about it. Okay? So what do? We go on and say, okay, let's now measure the response in that region in a bunch of new conditions. In this case, we wanted to answer a bunch of questions. We wanted to say, okay, does that fusiform face area respond to any body part? Maybe it's not just faces, maybe it's any body part. Does it respond to anything interesting? Maybe it's just engaged when you're paying more attention. And does it respond only to front views of faces or also side views of faces? Okay, so to do that, in the same scanning session, we ran another experiment where we had 3/4 views of faces with no hair. They look like criminals or friends and colleagues, and hands. And we gave subjects a task, a one-back task, where they have to press a button if any two consecutive stimuli are identical. That task is very easy for the faces and, frankly, very difficult for the hands by design. Okay? We don't go around comparing hands much or worrying whose hand that is. We spend a lot of time worrying whose face that is. So the idea was attention was focusing more on the hands than the faces. This is much more demanding of attention. And here's what so then what we do is we plot the response in that same region that we've already identified in each subject. And here's what we get. High response for the faces compared to the hands. Okay? So now, what we can say is that region that we've identified before as face selective doesn't respond to just any human body part, doesn't respond to hands. It's not just attention, because we drove attention harder on the hands than the faces. And it's not just front views, it generalized nicely to three-quarter views. Okay, so that's kind of a dopey little simple example to show you how you can use a functional localizer to directly answer a question like this. Okay? All right. So what are the benefits of this functional region of interest method? Well, first, it gives you a massive increase in statistical power. Okay? So I don't want to go on a whole sidebar about statistics, but if you just think intuitively, if you're doing a separate statistical test on each voxel in the brain, of which you might have, say, 50,000, then if you find that one of them shows a significant result, or 100 of them show a significant result at a p-level threshold of p less than 0.05, you should not be impressed, because you did a lot of tests. Okay, that's just sort of intuition, right? And so the way people deal with that problem is, if you do separate statistics on each voxel, you need a really extreme p-level in order to believe your result. So when you do a whole brain test, you have to get very impressive statistical significance in order to believe your result, because you're doing so many tests. One massive advantage of the region of interest approach is, instead of doing thousands of statistical tests, you're doing one. Or maybe, if you have a few regions of interest, 3 or 5, but not thousands. Okay? So that means you can use a much lower statistical threshold and believe your result. Okay, so there's a huge benefit in statistical power. Okay. Closely related, using functionally defined regions of interest ties your hands in a good way. Okay, so there's a lot of mischief in functional imaging research that comes from the fact that you have a shitload of data and many, many different ways of analyzing it. And if you troll through it long enough, you will find stuff, absolutely. And if you don't find it when you analyze it this way, analyze it that way and you will find something, right? And so there's just too many ways to fish around and find stuff in there. Okay, that's called degrees of freedom, lots of different ways to fish around, both because there's lots of voxels and lots of kinds of analyses. Okay, that's a problem. It means you can find stuff even if you started with pure noise. So if you use a functional region of interest, it ties your hands in a good way. It says, I'm not going to just look through everywhere, any which way, with every possible kind of analysis. It's like, I have a hypothesis. I have a question about a particular region, and I'm going to answer that question about that region in this rigidly determined way. And that means you're less likely to make up a lot of BS by analyzing noise. Okay? All right.
Is that a problem in the field?
It's a massive problem in the field. It's such a massive problem that I think a large proportion, I don't know if it's 30% or 70%, but a non-trivial proportion of published studies will not replicate because it has this problem. Rebecca knows more about this than I do. I'm going to go on. We should get back to that. How big a problem? Wait, it's a pretty big problem, isn't it? Yeah. Okay, and the next benefit of functional regions of interest is that you can get a nice measure of effect size, not just p-levels. Okay, what does that mean? I think part of the reason that the fusiform face area is so interesting is that that region responds much more to faces than anything else. If it responded, say, 2% to faces and 1.9% to everything else, I wouldn't have cared, even if that was significant. I think effect size matters in science, right? Not always. There can be some deep, deep truths about the universe that are based on teeny tiny effects. That can happen absolutely. But when you're choosing what to work on and which phenomena that turn up in your lab to chase after and which to ignore, I would pay attention to effect size. Okay, and also for lots of phenomena it matters, right? So imagine, a drug study does a study of a drug that's supposed to reduce the chance of heart attack. And they do this study on a huge number of people, and they find a very significant effect. P less than however many O's I put. That this drug reduces chance of heart attack compared to a placebo. But if the size of that effect is minuscule, who cares? Okay, so lots of times we care about effect size, not just significance. Everybody get that? And so the thing about a functional region of interest approach is that it gives you a very natural way to report effect size, right? We saw it right back here with this example I gave you. Here's a really nice effect size. That's honking, right? That's the technical word for this, honking, right? And you can just see it right there. It's not that color is just a P level. Like, what the hell does that mean? P-less and whatever, right? This is what we want to know, or at least that's what I want to know. I mean, it's got to be significant or you don't want to waste your time talking about it. But once it's significant, we want to know how big those effects are. Yeah? Okay. So functional regions of interest give you a very natural way to do that. Okay. Okay, so here's the more inchoate story about the benefit of functional regions of interest. And that is the idea that a functional region of interest identifies something that philosophers call a natural kind, a thing in nature that's a phenomenon we're studying, about which scientific principles can be discovered and that behaves in a law-like way. That's what science should be about. Science shouldn't be about coordinate location 1926 minus 57. That's not a thing we want to understand. The fusiform face area is a thing we want to understand. Okay? Many people will disagree with me on that. I'm just saying my bit, because I get to talk right now. Okay. And finally, as I mentioned a little while ago, this is also part of the same idea here. That makes possible a cumulative research enterprise, right? So if everybody's just reporting their telluric coordinates, we don't know how to relate those. Is it the same thing if they're like 2 millimeters apart? Is it not? Who the hell knows, right? But if everybody's using the same functional localizer to identify a region and then to say something about it, then we can all build on each other's results and have scientific nirvana where we actually get somewhere, right? Okay. All right, so those are the main benefits of functionally defined regions of interest. But I want to end by saying that actually it's not a choice, right? You can have the best of both worlds. There are many more sophisticated analyses that I won't go through in detail, but I'll just sketch something that I think is a lovely method that could be widely used and is not yet. And that is, and this deals in part with the situation where if you don't yet have a hypothesis about a region other people have talked about, what would your localizer be, right? So that's often the case. In the stuff I mentioned a while ago where we're trying to see if there are specialized brain regions for understanding the physical world, there was essentially no work on this. We were starting from scratch. We had no idea what we would see. And so what did? We did a group analysis. We did individual subject analyses and group analyses, right? But then to try to get our bearing in that, what we wanted to do when we started to find things that look systematic is define candidate regions of interest and then proceed. So here's something that you can do. You can do a hybrid analysis in which, I'm just going to sketch this very loosely to give you the gist. You split your data into two subsets. There are many ways of doing this. You could have most of it in one subset and only a teeny bit in the other. You could split it in half, say even runs, odd runs. Doesn't really matter. You split your data into two subsets. You use one subset to do a kind of soft group analysis, okay? So even when I described before that we couldn't find the face area in our group analysis, well, if we had really dropped the threshold or cheated in a variety of ways, of course we would see it. It wouldn't reach significance, but you could see it in various ways. Okay, so that's why I mean soft. You can do various versions of this that blur your data, lower your p-level threshold, do various things to discover a zone where you might find stuff. Okay, so now we have a zone in there. Now in each subject, we look for their activation that's in that zone that generalizes to the group. Okay? But now we've still only used half our data. So now we can look in the other half of the data and quantify the response, just like a normal region of interest approach. So what we've done here is essentially a group analysis, but by leaving a little bit of data out, by doing it in a soft way, where we're not leaning on the statistics in that original group analysis, by leaving some data out, we have huge statistical power and an unbiased measure of effect size in the left out data. And I'm not sure, but I suspect you can pretty much always do this. So there's a whole bunch of people who complain and say, well, we ran this study, but we didn't have a localizer because we couldn't fit that in our 2-hour scan slot. I submit that for any study where you would be doing a group analysis, you can do this and it will work much better, I think. Okay? All right. So, and there's some web stuff on this if you want to look at it. And that's it. Those are the reasons why I think functionally defined regions of interest make sense and how you do them and why they're not really the only thing to do. They're just a powerful complement to everything else.